So there are a lot of different strains of HIV. And there's different strands of HIV-1 and there's different strains of HIV-2. And we're going to focus on HIV-1. So we've got the major group, which is just abbreviated M, and then we've got these other two groups, outlier and then new. I know. And then if we break down M, they broke it into further letters, A, B, C, D, E, and so forth. And so you can be infected with, say, HIV, A or HIV, C. You can also get variations, right? So you can be infected with HIV-1 A/E, right? Because you can get a lot of genetic diversity in the HIV populations. This poses a really big problem for vaccine development. So the goal would be to develop one HIV vaccine that would protect against all of these different strains. Realistically, I don't know if that's possible. Different places of the world, you've got different strains that are more predominant. So in theory, maybe one day we might have like the North American vaccine, and then we could have the Central European vaccine, and then we could have the Southern Africa vaccine, and we could have the East Asian vaccine, and so forth. That ultimately, if we are able to develop a vaccine, that might be the direction that we need to go. Now, HIV is known for really replicating in our CD4 helper T cells and destroying our immune system. But HIV can infect other types of cells as well. Here's an electron micrograph showing HIV budding off of an infected cell. Here's the scale bars, so you can see each one of these particles is a little bit bigger than 100 nanometers across. We'll say 120 nanometers across. Alright, so HIV actually initially infects primarily macrophages and then goes on to infecting T cells. But HIV can also infect CD8 positive cells, NK cells, and it can infect some neuronal cells. HIV can sometimes infect nerve cells. Right? So primarily CD4 positive T cells, but it's not completely limited to those. So here's a cartoon showing the structure of the HIV particle. You guys should be familiar with the viral components in this figure. So we can start by talking about the envelope, right? So this is the outer layer. The envelope is stolen from the plasma membrane as the virus buds from the cell. Because it's stolen from the host membrane, right? So as the virus buds, it steals the host membrane. As it buds off and takes that host membrane, it's also going to have some host glycoproteins as well. So whatever components that were in the membrane when it bud off, it's taking those too. So we've got some host proteins. In the viral envelope, you've got the viral glycoprotein. GP just stands for glycoprotein, and 120 and 41, that's the weight of the protein. And so when they were first characterizing this, they're like, oh, it's a glycoprotein, about 120 kilodaltons. It's a trimer. So you can see there's 3 subunits there. It's a trimer. And GP120 is responsible for the binding. And GP140 is responsible for fusion and entry. And then we've got the matrix. The matrix helps with budding. And inside we've got the core. We've got the nucleocapsid, which is going to protect the viral genome. We've got the genome that's shown here with the little squiggles, right? It's an RNA virus, and we've got 2 copies. And then we've got reverse transcriptase, which is really important for making DNA from the RNA genome. And we've got an integrase, which is really important for integrating the viral DNA into your genome. The protease is important. The protease helps cleave the pre-proteins to make mature proteins. And then you've got a tRNA, a host tRNA. That's actually labeled right here, host tRNA. This attaches to the viral RNA, and this is really important. This is what the reverse transcriptase actually binds to start reverse transcription. And then you've got another host protein here, CYPA. This binds to the nucleocapsid and helps stabilize it, and potentially helps protect the nucleocapsid from defense mechanisms inside the cell. But people are still trying to work out exactly how this protein works. We know if you remove it, it makes the virus less infectious. But people are still trying to work out exactly how this helps the cell, if it just stabilizes the nucleocapsid or if it does other things too. Okay, so first if we look at entry, so we've got the viral envelope here. And we've got our glycoprotein, GP120 and GP41, in the viral envelope. Now, first, the GP120 binds to CD4, and then, and this is in the cell membrane. This is not the endosome. This is on the cell membrane. So CD4 binds to GP120. And that brings the virus kind of really in close contact with the cell. And then you need a co-receptor. So this here is a CCR5. So this is a protein that helps an immune system function. This binds chemokines. But the virus uses this. You need to have binding first to CD4, and then you also bind to CCR5. So after GP120 binds CD4 and then CCR5, some conformational changes happen, and that exposes the fusion peptide in GP41. So GP41 has a fusion peptide at the end. This gets inserted into the plasma membrane of the cell, and then it folds back upon itself. And this allows the fusion of the viral envelope with the plasma membrane of the cell. And I'm going to show you a short animation of this here in just a second. I don't know why this video doesn't have sound, but it doesn't. So the virus comes along and it sort of surfs along the surface of the cell. Remember that membranes are fluid mosaics, right? So things, proteins, glycoproteins, cholesterol, you've got all of these things in the membrane that can float around and move. And since the viral envelope is just a lipid bilayer that it stole from the host plasma membrane, the glycoproteins, the envelope proteins in the viral envelope, can also float around and move in the envelope. So you get enough together to firmly grab the virus and bring it in really close proximity with the cell, and CD4 binds, and then the co-receptor binds, and then that's the fusion peptide of GP41 that inserts into the host cell membrane. And they're just showing one of these for simplicity's sake, but you'd get several of these complexes doing the same thing at the same time. And GP41 folds back upon itself. And then that allows for the membranes to fuse. And then the core of the virus gets dumped into the cell. So GP120 is responsible for binding and attachment, and GP41 is responsible for fusion and entry. If we look at the HIV viral genome, we've got several different genes, several different gene products that we need to think about. The regulation of gene expression is similar to some of the things we've already seen. We're going to have our early, middle, and late genes. In the genome, you've got your long terminal repeats at either end. We've got the GAG proteins. This is going to be our matrix and nucleocapsid. We've got pol. So these are going to be associated with polymerase. So the reverse transcriptase. You've also got protease and integrase. And then the envelope, right? We've got our GP120, GP41. And then we have some proteins that are expressed through alternative splicing. So, for example, some of the other associated proteins, like TAT, for example, and we'll talk about that, are produced through alternative splicing. Now, the virus is going to come in with 2 copies of its RNA genome, and it needs reverse transcriptase to run off DNA. So reverse transcriptase is going to bind, as I mentioned, to the host tRNA that is on the viral RNA, and the reverse transcriptase needs to bind to that to start reverse transcription. So after reverse transcription, the DNA is flanked by the terminal repeats, and the 5' terminal repeat is a promoter sequence. The viral DNA is then transported into the nucleus, and integrase inserts the viral genome into the host genome. So this is the most important step. A lot of people focus on reverse transcription, which is cool and is important and is unique, and kind of blew away the idea of central dogma, right, that you can make DNA from RNA. So reverse transcription is cool, but without integration, we wouldn't have our retrovirus. When the viral RNA gets converted to viral DNA, and the viral DNA gets integrated into the host genome, that's when you're infected forever. After that integration event, the virus is now part of you, and at that point, you can't be cured. At that point, you're going to be infected forever. Now, there are good treatments, but if you stopped taking the treatment, you would get sick again. After the integration, the virus can be latent, or it can proceed with active infection. The fact that you've got a lot of cells that are latently infected is one of the biggest problems, or I should say the biggest problem to elimination of the virus. Right? You can be on treatment and there is no detectable virus in your blood. As soon as you stop treatment, the virus will come out of latency and start replicating again. So in order to have efficient transcription, we need a couple regulatory proteins. The first one that we're going to talk about is TAT, or the trans-activator of transcription. So TAT is really important. It binds to TAR. TAR is the trans-acting response element that is in the viral mRNA. So if you've got viral mRNA, in the viral mRNA, you've got TAR. And TAT has to bind to TAR in order for transcription to be efficient. And so remember, the virus uses reverse transcription initially to make the DNA, but after it gets integrated by integrase into your genome, you're now using the host RNA polymerase. So having TAT attached to TAR really increases transcription by the host RNA polymerase by like 1,000-fold, right? So TAT is really important for efficient transcription of the viral genes. And then we've got REV. And REV is really important for translation, protein expression for translation of the unspliced messenger RNAs. So REV is going to bind to RRE, which is the REV RNA element. And so REV, again, is going to be a protein. If we've got our viral messenger RNA, then we need REV to bind to that, and this allows for an increase in export to the cytoplasm, and this increases translation. So remember that after integration, after integrase, now the virus is dependent upon your RNA polymerase, some RNA polymerase 2, and the virus is dependent upon your ribosomes in order to get its genes translated. So HIV has a lot of control elements, or a lot of regulatory elements. The ones that we're going to focus on, or I'm going to specifically mention again, are the long terminal repeats, and these are really important for transcriptional promoters. So this has the promoter elements for cellular transcription factors. So the long terminal repeat, especially the 5' long terminal repeat, has important promoter elements. And then we've already mentioned the TAR and RRE. The other regulatory elements just mentioned, again, these are important for binding to cellular transcription factors and regulating the course of HIV replication. So a couple other important gene products, NEF, VPR, VPU, and VIF. First, let's remind ourselves that HIV evolved from SIV, and it had to jump species. And our cells have a defense mechanism against viruses called tetherin. And tetherin can bind viruses as they're attempting to bud off, and then hold on to them so they can't leave. And then your immune system could come along and it could tag these with antibodies, and this could lead to antibody-dependent cell-mediated cytotoxicity, and just being able to limit the exit, the egress of the virus can limit the course of infection. So a successful infection by HIV or SIV has got to find a way to antagonize or block the activity of tetherin. And different strains of HIV or SIV have had different ways of doing that. So all SIV and HIV have to get around tetherin. And if we look at how we were managed to jump species with HIV-1 and HIV-2, there were a couple different ways of doing that. Now, when we just had simian immunodeficiency virus and it was circulating in monkeys, either VPU or NEF was able to antagonize tetherin, but the tetherin is slightly different in monkeys than it is in the great apes, and so to jump that species barrier, the virus evolved a different way of doing it, and it had some mutations in NEF, and now NEF antagonizes tetherin, and they do this by slightly different ways. So that's shown in figure B here. So VPU interacts with the transmembrane region of tetherin. NEF interacts with the cytoplasmic region of tetherin, but both of them stop tetherin from working. So in the great apes, NEF was responsible for antagonizing tetherin. Now, when we jump species again into humans, they had to have a different mutation because humans have a slightly different tetherin sequence than the apes did. So another set of mutations was required for this. So some mutations in the major group here of M, some changes in VPU, and in HIV, actually the envelope protein. So this is a GP120, GP41. The envelope protein actually gained the function, some mutations occurred, and it gained the function of antagonizing tetherin. So we can track down the specific mutations and follow the progression from monkeys to apes to people. If you want to know the details of this, if this is something that's cool and interesting to you, this paper where I got this this figure from and one of the other figures in the presentation has also been uploaded to Canvas, so you guys can check that out. Now that covers NEF, VPU. I want to talk briefly about VPR and VIF. Right. So VPR, this is a viral protein R, is kind of a rock star protein here. Like a lot of viruses, you sometimes need one protein to do lots of different things. VPR is really important for the replication of the virus, for regulation of the virus, for bringing it out of latency. VPR can have different forms. It can be a monomer, it can be a multimer. VPR, when the virus enters the cell, VPR goes to the nucleus to help with replication. VPR is also exported, and it can act in an endocrine function. VPR can cross the blood-brain barrier, and we think it can lead to some neuropathology in advanced infections. VPR, when it's extracellular, can lead to apoptosis. So it leads the membrane to be permeable to cations like calcium and magnesium, and then that triggers the mitochondrial pathway. So the mitochondria starts releasing pro-apoptosis factors, triggering cell death. You guys might want to go back to the earlier chapters and review the process of apoptosis. So within the virion, this protein VPR is very, very concentrated. There's over 700 copies of VPR in each virion, so it's a really important protein. It can stop T cells from proliferating, and then it triggers those T cells to differentiate. This makes them better hosts for HIV. It stops the cell at the G2 checkpoint, and this is when transcription from the promoters in the 5' LTR are most active. So VPR does a lot of things for the virus. Other proteins, here you need to talk about the last one I'm going to mention here is the virion infectivity factor, or VIF. So we mentioned that NEF and VPU are important for antagonizing tetherin, which is a host defense mechanism. So VIF is also responsible for antagonizing host defense mechanism. So VIF, I was trying to switch colors there. VIF, all right, we'll be in purple now. VIF antagonizes ApoBec. So this protein, it's actually a protein complex. ApoBec 3G. I'm going to move to a smaller. ApoBec 3G, this attacks the viral genome, and this leads to a lot of mutations from C to U, ultimately going from G to A. So this is a deaminase. So if we've got, hold on, I can draw this better. Okay. All right, so if we've got cytidine, right? ApoBec deaminates it and turns it to uridine. So we got our and now C always binds to G and U always binds to A. So if it makes this change in the RNA, then in the DNA, instead of this leading to a G here, it leads to an A. So this changes in the RNA cysteine to uracil, so in the DNA it's going to go from guanosine to adenine. And so you get a whole bunch of these mutations. And you get an overload of mutations, and then the virus can't function anymore. So that protein, again, was ApoBec. ApoBec. And ApoBec 3G. And VIF antagonizes this. So we've got a couple defense mechanisms here, right? So NEF and VPU are going to antagonize tetherin. VIF is going to antagonize ApoBec, and then VPR is just the all-star protein that does lots and lots of things for HIV infection. All right, we're going to move on. And just in summary here, so we'll kind of run through all this again real quick in summary. So the virus is going to use GP120 to first bind CD4 and then CCR5, and then GP41 is going to lead to fusion. And then once the core gets into the cytoplasm, it uncoats, reverse transcriptase is going to bind, it's going to start from that tRNA that's on the viral RNA, and we're going to undergo reverse transcription. So now we've got DNA, we've got viral DNA. The viral DNA is going to enter the nucleus. Integrase is going to integrate it into the host genome. At this point, it's a part of you forever. After integration, some cells are going to remain latent. Some viruses are going to undergo active lytic infection. So we're going to use the host RNA polymerase to make viral messenger RNAs. The host RNA polymerase is dependent upon transcription factors that are in the 5' terminal repeat, the 5' LTR, and it's going to start making our viral messenger RNAs. Some of those messenger RNAs get spliced, some of them don't. In order to increase the rate of transcription, we've got a couple factors. We mentioned in particular that we've got TAT. So TAT is going to help increase transcription. And we also mentioned REV, which is going to help move unspliced messenger RNAs into the cytoplasm. Regulation of transcription, regulation of gene expression is going to be broken into our early and late genes. So first, we've got to start making more TAT and REV, because those are going to help you get more transcripts made. That's going to get the ball rolling, and then you've got to get more of your structural proteins made later on. So some proteins are going to be translated on free-floating ribosomes in the cytoplasm, but the envelope protein, so it's going to start off as actually a GP160, because that's 120 plus 40 essentially added together. So the glycoprotein is going to get translated on the rough ER, and then it's going to go through the Golgi. In the Golgi, it's really important, a cellular protease is going to cleave GP160 into GP120 and GP41. If that cleavage doesn't occur, the virus is not infective. So the glycoprotein gets cleaved and it gets glycosylated, so it gets decorated with sugars. Those sugars are also important for antibody recognition or lack of antibody recognition. And then those glycoproteins move to the plasma membrane, where they wait for all of the other components to come together. So all of these other proteins have to get made. You've got to make more nucleocapsids. You've got to make the matrix protein. You've got to make your VIF and VPU. Remember, the VIF is going to help antagonize ApoBec, and VPU, that's going to be our protein that's going to help antagonize tetherin. And remember, VPR is going to do lots and lots of things for the cell. It's going to, not for the cell, for the virus. It's going to attack the cell. It's going to lock the cell. It's going to stop it at the G2 checkpoint, which makes the promoters in the 5' LTR, that's where they're most active. Remember, VPR can be secreted as well, and when it's secreted, it can lead to the cell death of other cells. It can permealize membranes to calcium, and it can trigger apoptosis. So VPR has lots of functions. All of these proteins are going to meet up at the plasma membrane. The matrix is going to help trigger fusion, and then tetherin is antagonized so the virus can get released. Another thing that to note, just real briefly, Another function of VPU also causes a downregulation or a reduced expression of CD4. And that also helps the virus bud off, because if this cell was producing lots of CD4, then it could attach to these virions as they're leaving, just like we talked about with other viruses, right? Influenza virus has neuraminidase to cleave sialic acid, so the new viruses don't get stuck to the old cell that it's leaving. Now, HIV starts off with a preference for CCR5, but as the infection in your body progresses, the tropism changes, and it starts preferring CXCR4. This is a co-receptor that's expressed more highly on the helper T cells. So initially, the first viruses that infect you have a preference for using CCR5, but as the virus evolves in your body, it develops a preference for using CXCR4. And we'll talk about that more in just a minute.